In this video module, we're going to take a constitutional sample that we generated using the single sample analysis in the analysis workflow and show how we can query the ChAS database to help put annotations to the sample itself as well as individual segments. First, we're going to review histograms, which is a representation of the segments that are currently in your database, and how to filter the histograms using certain aspects which are applicable, such as sample type or call. We will then show how to add sample level annotations, such as phenotype or interpretation. And then using what we call the segments intersection view, which will show the segments that are in your database intersect with your segment in your current sample. How to add annotations to the individual segments. And then how we handle mosaic segments in the ChAS database. And lastly, publishing these segments to the ChAS database. So before we go into the software, we want to review just a couple definitions that we're going to be talking about quite a bit when querying the ChAS database. So in order to bring back segments from your database that are of relative size to your current segment in your sample, we use something called overlap and coverage. So our top segment here is the segment in our current sample, and we're interested in seeing what in our ChAS database have we seen before that's relative in terms of size to this particular segment. So we're going to use something we call overlap, and as well as coverage in order to bring back samples of relative size instead of bringing back whole chromosome or whole arm, which may not be applicable to a segment of this size. So for instance, in our database, if we have segment A and segment B, we're going to look at the overlap of my segment with segment A. So my segment overlaps 100% of segment A. So my value would be 100% for overlap. But in terms of coverage, segment A covers my sample by about 20%. So my value is going to be 20% coverage. Whereas for segment B, which is a very large segment, my segment only overlaps about 30% of segment B. But segment B overlaps 100% of my segment. So that would be the value assigned to the coverage is 100%. So the default in the software will bring back segments that match both a 50% overlap and a 50% coverage. So in this case, neither segment would be returned. Now you can adjust the overlap and coverage thresholds to meet your criteria. You can make them more or less stringent depending on how you see fit. Let's go ahead and analyze some files. So I'm going to log in to the ChAS database server on my laptop using the default that came with the installer, admin. Click OK. And now the first thing I want to talk about are the histograms. So we have the default histograms showing LOH, gains and losses represented in my particular database. I'm going to go ahead and turn off the mouse overs for the demo. So I go under View and Mouse Over pop-ups. We'll just get rid of that for now so it's not distracting. So you can certainly change with the way you're viewing the histograms. We have different options. So we can go here to my default histogram, right-click on it, and choose View Edit Properties. You have a couple options here. Under the Histogram Filters tab, you could rename it. You can show the axis values, which I am, so that you see what's going on the y-axis. You can show a grid line if you'd like, which will make a grid lines horizontally. And then solid bars is what I have selected as to the way we're viewing the filled in graph down here. You can also choose not solid bars, which will appear as hollow boxes. For the y-axis, you do have options for dynamic range or fixed range. I have my fixed range set from 0 to 20. If you do select dynamic range, for instance, in my LOH graph behind here, you see I have about segments in this particular region. And so therefore, the y-axis would be set dynamically from 0 to 4. You also can turn off any of the histograms that you may or may not want to see. And we're going to get into histogram filters in just a second. So we'll come back to this screen and say OK. So now you can see the grid lines running horizontally. So now we can take, and if we're interested, this gives us a nice representation of what's going on in our database and where we've seen segments before. So I'm going to go here to chromosome 14, where we have our nice little spike here to about 9. And I'm going to right click because I'm interested in knowing what I've seen in this particular region of the histogram previously. So I can right click and say show histogram items in that bin. When I right click, I get this segment intersections view, which we're going to visit quite a bit in this tutorial, and pull down my menu. I can see the samples that I have seen before in this particular bin. It gives me the copy number state, the chromosome min and max, as well as what I've called these particular segments before in the other samples, as well as any interpretation that I've set with them. So I can see I've called them, this is the IGH region, and I've called them benign. I can zoom in and zoom out to see the segment size in this particular region. These vertical blue lines are the region of the histogram that I was clicking in, but you can see the size of the segments that cross over into that histogram. If I briefly turn on the mouse overs again, you can see as I mouse over the histogram, I get a quick look to see, okay, in this particular region of the histogram, under bin value, I have 9 samples, which is 36% of the samples that I have loaded in the database. So in my current database, I have 25 samples. So this is everything in my database. I have both constitutional and cancer samples in my database. So perhaps I just want to look at constitutional samples because that's what I'm going to be viewing today. I'm going to be looking at a constitutional sample. So I can go up here to the ChAS database and choose Add a Histogram. In this case, I'm going to look at all samples in my database that are just annotated as being blood samples. So I'm going to call this my blood histogram. I'm going to use my fixed range still, and I'm going to make it from 0 to 10. And I'm going to take off the grid bars. I'm going to filter by blood constitutional. So any sample that has been annotated as blood constitutional is what I'll see, segments relating to any sample associated as blood constitutional. You also have the option of filtering just on females if you wanted to see just females. Or also, if you wanted to show regions that just have pathogenic calls, you can do that as well. We're going to go ahead and just use blood constitutional as our filter and say OK. And now if we look to the far left here, we have blood histogram. So a brand new set of histograms. If I zoom up here a little bit. We have the default histogram, which we were viewing before. And now we have this new blood histogram, which we can see now. For instance, in LOH, it filtered out these samples that were LOH along the length of the chromosome, because now we just see our one small segment that was in a sample that was annotated as blood. So since we're looking at a blood sample today, I'm going to go ahead and turn off just by clicking my default histogram so that we're just looking at my blood histogram. And you can add as many histograms as you'd like. You could have a cancer histogram, or again, a pathogenic call histogram, or ones by gender. All right, before we load the sample, we're going to go into just a little bit of what we talked about of where you can set your overlap and coverage count. We're going to click on this notepad or the edit configuration dialog box. And so we have the tabs for those who've used ChAS before that we've seen in terms of the segment data, and if you want to use smoothing and joining, setting any sort of QC thresholds, color rules for your AED files. But what I'm interested in here is going to the FAR tab right now, which is the database query tab, which is new. So the default is 50% coverage and 50% overlap for copy number, as well as LOH query. Again, you can change these to make them more or less stringent. You also have the option of, if I'm looking at a gain in my sample, if I have this checkbox checked, that means only bring me back gains. I'm not interested in seeing losses in that same region. I'm only interested in the same like. So if it's a gain, just bring me back gains. If I'm looking at a loss for a sample, when I query the database, just give me losses. In this case, we'll uncheck that because I'm interested in anything copy number that matches my 50% coverage and 50% overlap. And I also have the option of saying, also bring me back LOH. I can uncheck that to say when I query a gain segment, only bring me back copy number because that's all I'm interested in. But if I'm interested in anything that matches that region at all, whether it be copy number or LOH, I can check the include LOH button. I also want to talk a little bit about vocabularies before we move on, because a lot of this module we're going to be putting annotations with segments. So we have some segment annotations, which would be call, and the default values are pathogenic, likely pathogenic, unknown significance, likely benign, and benign. You can certainly add your own custom call and add it to this list so that it can be selected. So I'll just make one not clinically relevant and add it. Okay. So now we have interpretation as well. So this would be interpretation for this segment. I can add something like maybe it's an unbalanced translocation. So now these would be, instead of my typing every time I see part of an unbalanced translocation, I can just right click in that cell, and we'll walk through how to do that, and choose unbalanced translocation. A new field for this version of ChAS is the inheritance tab. The default is de novo, maternal, paternal, and unknown. I've previously added SIB, but again, you can add anything that you want for this as well, this field as well, to be part of your dropdown. Now the last two have to do with the sample type itself. So the default list goes through fixed pellets. Some of these others are cancers that I have added. And again, same as you would for the call field, these are going to be available in a dropdown. And you can choose, let's see, like lung tumor. And what's nice about these is that it will help you filter your data, like the histogram filter that we just walked through. So you can really focus your histogram analysis and your analysis on the relevant sample type. And then the last one is phenotype. This phenotype list comes from and is organized similar to what you would see on the HPO site, the Human Phenotype Ontology site. Again, you can add more phenotypes as you see fit. Okay. Let's go ahead and add some samples. So I'm going to do File, Open. I'm going to go to my ChAS 3 data and choose the constitutional blood sample that we analyzed earlier and a cancer sample. For right now, I'm going to turn off the cancer sample, and we'll come back to that. Right now, I'm going to focus mostly on this constitutional sample. So I also want to load some AED files that are available to you. One is going to be this developmental delay genotype to phenotype AED file, and also a common artifacts bed file as well. There is a video module on cytoregions which will give you more details, so please refer to that module on creating the cytoregions file. What I'm going to do is go ahead and make this decipher file my cytoregions file. And just as a quick reminder, it's just a very similar to a bed file, but we call it an AED file, and it is genomic coordinates of regions that are known to be clinically relevant, and I'm interested in focusing in on. So I assign it my cytoregions file, and I get these gray bars highlighting those regions. Now the common artifacts, we're going to assign that as our overlap map file. And the overlap map file tells me, I'm interested to know that my segment of my sample overlaps with this particular region, but it's known to be just an artifact. Maybe it's a segmental duplication region, and it comes up a lot, but it's not clinically relevant. And so by assigning that as my overlap map, it will jump out and just make it easier for me to assign annotation. Okay, so now we have our sample loaded, and the first thing I want to do is go ahead and add sample level annotations with this particular file. So I'm going to right click on my file name and choose View Edit Properties, and come here to the Sample Properties tab. Maybe if you have a lab ID, you don't have to fill this in, it's optional. You can even name it the same thing as the chip file, or if you have a lab ID that you wanted to use, you could type that in here. The sample type is what I get from my drop-down list that we saw in the Edit User Configuration Sample Type tab. So this one's blood. And now phenotype. In this field, I can do a free text and say this one had microcephaly, or I can take and right-click in this field and click to select, and this is where I get my long list of phenotype options. And so I'm also, let's say, cleft lip, and say, how about, we scroll through our list, learning disability. And again, you can either free text it, or instead of having to type that each time, you do have the option of using a snippet by right-clicking in that field. This interpretation is going to be meant for the sample level interpretation, so since we haven't looked at the sample yet, we'll leave that blank and we'll come back to it later. Say OK. And then I just want to make sure that my filter settings are what I would like them to be, so I'm going to use my cytoregions analysis settings. And again, there is a video module on how to set up your filter settings, so please refer to that, or the user guide on how to create your filter settings. All right, so my filter settings are looking at 50 markers and 50 kb for gains, and 25 markers and 25 kb for losses. So as I look at the Cario view, I can see I do have a few segments. So we're going to go ahead and go to the segments table and start diving into these segments individually. So I see I have a total of 17 segments that meet my criteria. Just for now, I'm going to turn off the LOH data type, which is down here, and just focus on copy number. I'm going to scroll over, so I have certain columns that I'm interested in seeing. I have chromosome, just the size, the marker count, OMIM genes, but I'm going to start right here because this is the region that I'm going to interact with the most. And when you're setting up your segments table, this little icon over here, you can select columns or turn off columns, whichever ones you would like to use. And also you can drag and drop just by holding it down. You can drag them into the order that you would like to see them in your segment table. Okay, so the first item, I can click on the first row, and I can see I do have a segment here. It's a dark red showing me that it's a copy number state of 0. I can look down here, my histograms, it looks like I've seen it before. I can tell by my overlap map, this does overlap with something that is known to be an artifact by 100%. But if I didn't have that, I can also come over here to my database count both column. What that means is there's 5 segments in my ChAS database that match both the overlap and the coverage criteria. You can also look at the segments that meet just the overlap, or just the coverage, or just the overlap. But both, the dB count both means it overlaps, it meets the thresholds for both overlap and coverage. So that's the one I'm going to focus on the most here. I'm going to right click on it, this particular cell, and choose Query ChAS DB for dB count both. So what I can see here is, here's my query segment. This is the segment in my current sample. These are the segments in my database. I have the vertical blue lines so that you can see the breakpoints and where the intersections are across these segments. And now I can see the file name, as well as the chromosome start and stop, any sort of call associated with that segment, or interpretation. And if I scroll over, I get the values for the coverage and the overlap, as well as the sample type. So these were all blood samples, and any phenotype that was associated with that given sample. And again, just like the segments table, there are other columns that you can add. And so just by clicking on this particular icon, you can check off if you want to see the cytoregions, for instance, and that will be added to the end of the column. Okay, so in this case, I see that I've seen it several times before. I have called it benign, and I do know that it does overlap with an overlap map item by 100%. So, all right, I can go ahead and I feel confident calling this benign. My interpretation is that it's we'll call that an artifact. And I don't have any inheritance information for this particular one. Okay, I can move on to the next one. So again, I see this is another region that overlaps with my overlap map. I have seen it several times in my database. So if I want to, I can click in there just to see. And again, yes, I've seen it several times. I can see the sample names for all the times that I've seen it, as well as I've called it benign every single time, and this is part of the IGH region. Okay, great. I feel confident. I can call this benign and IGH. Okay, here's a region on chromosome 20. It doesn't hit either my cyto region or my overlap map, and I've never seen it before. So this is when we would start digging in and using the genes track and DGV to see if we can figure out what's going on here. I do see a dip in the log too, so this is quite believable. There's a lot of markers in there. If I turn on the genes track, let's expand this back out by double clicking, I can see I do overlap with a couple genes. Let me see about the DGV. Let me also turn on the database of genomic variants track and see if there's any overlap. I'm going to go ahead and expand this since I do see 2 tracks to make sure that I'm not missing anything. All right, so there is a loss, but it doesn't completely overlap my particular segment. So I'm going to go ahead and collapse that back down and turn that track off to save space. What about OMIM? Does it overlap OMIM? Does overlap a couple OMIM genes. They are gray, which means they don't have a phenotype associated with them. So they're not as part of the deciphered DDG2P track, but I think they're probably benign, so I can call them likely benign. They do have an OMIM gene reference, but I don't think that's what's going on in this particular case. Okay, let's go on to the next one. So here on chromosome 22, I have a couple different segments. Let's go ahead and make this a little bit bigger so we can see it. When I'm looking at the track, so the copy number algorithm has called some segments as well as the mosaic copy number algorithm. In this particular case, it doesn't look to me like it truly is mosaic. This looks like a loss. I don't see a divided head track in this particular region. So we're going to use the edit function to clean this region up a little bit. For more information on modifying segments, there is a module on segment modification, so I do recommend viewing that. But in this case, we'll review it briefly. So I'm going to go into edit mode. And now go back to chromosome 22. Okay, so I'm in edit mode. In edit mode, my mosaic segment now is highlighted. I can see the light turquoise highlighting around that particular box. I'm just going to go ahead and get rid of this segment because I don't believe it is a mosaic. So by right-clicking in that segment, I can say delete this segment. And am I sure I want to delete it? Yeah, I'm sure. You can always undo it and get it back, but I don't believe this is truly mosaic. I believe the copy number segments. And so I'm also going to take and edit my copy number segments to reflect the probe coverage. So in this case, I'm going to highlight all three of these losses. You can see my 3 tracks up above get highlighted. I'm going to merge these into one segment. I don't believe these are actually 3 individual segments. So I'm going to merge them together. It's a loss of copy number state 1. And I'm also going to take it and edit the breakpoint to start where the probe coverage really starts. This primarily looks diploid, so I'm going to go ahead and start the segment here. So if I highlight that segment, choose the double-headed arrow, and now I can look for a double-headed arrow as I grab that shadow box and bring it to, and it will snap it to the nearest probe. So that just looks a little better to me. I just feel a little bit more confident in that. Okay. And in terms of this end, I'm okay with this end. So we'll hit the double-headed arrow with the green check. And great. Now, again, with this second segment here, this truly does look like a loss to me, but this side of it looks more diploid. So I'm going to edit the breakpoints on this one as well. So I'm clicking my double-headed arrow, grabbing the double-headed arrow and bringing it back to this region where what I believe the breakpoint is, and then hitting the green check. Okay, so I've cleaned up this area a little bit to reflect more what I'm seeing in the data. And now I'm going to go ahead and get out of edit mode. And now continue on with putting annotation with my segments. So I can either click on this segment itself and it will highlight the row. Now I can see that there is a cytoregion, so from my decipher file down here, that does overlap with my particular segment of a 22q11.2 deletion. I've never seen it before in my database. And so, but I do have 4 segments that have covered this area. So let's just look at that for a second to see what we're looking at. So when we meet the coverage threshold and we zoom out, we can see what coverage does, right? So we have this smaller segment, and then we have these much larger segments, and that's why you don't see anything. You see a 0 in the database coverage count, both, because this segment is relatively small compared to the others. I do see a segment down here that is quite interesting to me. It is a little bit bigger. It does seem to be to overlap quite a bit with my segment. So let me go back up here and look. And so I see a database overlap of 48%. So it's just under that 50% cutoff. So that's interesting to me. And this one was interpreted, this segment, as pathogenic with a 22q11.2 deletion. And if I ever wanted to go back to that sample, I have the sample name here. So okay, that's interesting. I do have, it overlaps my cytoregions, and I have seen a sample that is similar. Again, I can go back here and go to my segment table, and I can even filter this down again. So I've seen the overlap be 48%. So you know what? I want to filter my search criteria again. I also happen to see a couple samples here that are renal cell, which are cancer. So they're not applicable to this particular analysis. So I'm going to filter my data again. So I will go ahead and leave it on gain loss and LOH, but I want to filter my coverage to be, let's say, 40% and my overlap 40%. My sample type is blood, and say OK. And now I come back with two segments that meet my criteria. That same segment we were talking about before that has been labeled as pathogenic, and also this LOH segment as well. So we have a 48% overlap and a 99% coverage for this sample. And so now it meets my criteria. So now we can go back. All right, we're feeling pretty good about this now. We've seen it not only in our CITO regions, but also in our database. We'll call it pathogenic. And this is going to be a 22Q11.2 deletion. Okay, I do have the next segment on 22, which is overlapping two regions in my CITO regions. It is overlapping quite a few genes, as we see down here in the gene track, and quite a few OMIM genes as well. So we can go through and either call it, we haven't seen it before in our database, so we can call this pathogenic, or we can go through and actually do a little bit more in-depth analysis on these two syndromes that are in our decipher track, and we will see that those are autosomal recessive, and so they may not be applicable to this particular case. But more research would have to be done in the literature to really define what that should be called. So in this particular case, just for our demonstration purposes, we'll go ahead and call it pathogenic. And then we can call it the two different syndromes. All right. So as we continue on, again, we have another region here that we haven't seen in our database before, but it does overlap 100% with a known region that's artifactual from our overlap map. As we can see at the very bottom here, it matches our overlap map. So we can feel pretty good about calling that benign. We'll go on to the next one. We have this region on 8, again overlapping with a known region that comes up quite a bit. We've seen it 10 times in our database, so let's go ahead and right click and just for fun check. Oh yeah, we've seen it quite a few times. The breakpoints are fairly consistent across all our sample. We zoom out and we can get the names of any of the samples that we wanted to. If I want to know, okay, well this one sticks out a little bit further, I'm interested to know what sample that is, I can simply click on that segment and it will highlight it in the row above. Oh, okay, that came from this particular sample if I wanted to go back and follow up some more. This was called benign quite often, and given the interpretation of the Atom 3A area. But again, if you wanted to filter down even further and say search again, I'm interested in searching on things that are just blood and been called benign. Then you can say OK, and your data will be filtered based on just the call and the sample type. So we are just seeing things that are called benign now. This filtering, the secondary filtering that we're doing, does not affect the overall filtering that we set on the configuration tab for overlap and coverage. It's just for this little instance when we want to drill down a little bit deeper in this particular region. Okay, we feel pretty good about that. We're going to call, just click to that. That one was benign. We're going to call this one benign as well. and atom 3A. Okay. All right, we're down to our last one on chromosome X. Again, we haven't seen it in our database. It doesn't seem to cross any genes, and there's nothing in terms of decipher. We could go through and put on the database of genomic variants. We do see some variants in this particular region, and it doesn't cross with any genes, so I'm just going to call this, it's just not clinically relevant, or you can call it benign. All right, we have analyzed all the copy number segments in our particular sample, and in this particular case, due to what the phenotype is that was given to us, we are going to go ahead and annotate this particular sample as the 22q11.2 deletion. So what I can do is come here to this particular array nomenclature and do Control C. I'm going to copy that and go back to my constitutional blood sample and right click on it so that I can add this information to the sample interpretation field. So choose View Edit Properties, Sample Properties. Now remember, this is where we put in the phenotype. We're going to Control V and paste it right there. So there's the array nomenclature, and then this is free text. You can add any sort of information that you want to this. So micro deletion 22q11.2. Okay. So we've added the sample interpretation, and now we can publish samples to the database. I want to talk about two things just to keep in mind. You cannot publish in edit mode. So like we were in edit mode when we were deleting some of the segments and some of the mosaic segments and merging others. If I go to then publish by either right-clicking on the sample, you can see publish files to database is grayed out. Or this icon up here is also grayed out. So you cannot, it's just a safety feature, you cannot publish in edit mode. So if it's ever grayed out, you can go, I must still be in edit mode. So go ahead and turn off edit mode. And now if you highlight that sample, you can see that that icon is now active. So we'll go ahead and publish. So now it's letting me know, you get a quick warning saying that the gain is enabled and filtered. The loss is enabled and filtered, so that's great. But if you remember, we turned off LOH. So I turned it off because I just was going to deal with the copy number. So it's reminding me, are you sure you want to upload? Because LOH is not enabled, so meaning it's not turned on here, so therefore it will not be published. Therefore, it gives me the opportunity to go back and look at the LOH tracks to see if I would like to go ahead and this one's due to a loss, so that's fine. I don't care about uploading it, and I can go through these other LOH tracks to see if I'm interested in uploading them or not. So in this case, I'm not going to. I'm going to turn that off and then go ahead and publish my file. You will see that the mosaic gain and mosaic loss are not publishable, and we're going to talk about that in our second sample here as to why that is. So it's just to let you know that you can't publish them. It's sort of a reminder to go back and address the mosaic segments that you have in your sample. So am I sure I want to publish? Yes. All right, I have been successfully published, and the database count, overlap, and coverage in both segments, columns, are automatically updated. As you can see, remember they were all zeros here before, there are now ones. That's automatically updated. The histograms have to be regenerated manually. That was done on purpose. For larger databases, it takes a few seconds, up to a minute or so, for the histograms to regenerate. And so rather than have to wait for that each time, that at this point is a manual step, but you can just come up here and say refresh my ChAS DB data. It's fairly quick on my laptop. as I have only about 30 samples in my database, but as the database gets bigger, it will take a little bit longer for those to regenerate, so that's done manually at this point. You can republish data. So let's say in my constitutional sample that I just did that I have found that this particular region, which does have some OMIM, we found later on some data that suggested that these actually are clinically relevant and it's likely pathogenic. So you wanted to go back and edit that particular information. You can now choose likely pathogenic, right click, and choose publish the file to database. It's giving me that same message. Now it does recognize that that sample is already in. Are you sure you want to publish this and overwrite the data? Yes, I do. I want to update it. So now if I go to query that particular region, it will say likely pathogenic. So we saw how to go to the segment intersections window using the segment table by right-clicking on the DB count both column. You can also click on anywhere in the table and choose query ChAS DB. You'll notice it says query ChAS DB as opposed to query ChAS DB for DB count both. So this is the only column that you can right-click on to get segments that meet both the overlap and the coverage thresholds. If you click anywhere else in the table and query ChAS DB, it will bring back any segments that match either the overlap or, as you can see up here, or the coverage. So just keep that in mind when you're querying the database. You can also query directly from the segment itself down here, but again, it's going to give you overlap or coverage thresholds as opposed to both. And then same with the database, same with the histogram. You can also get to that segment intersections. Whether you have a segment or not, it's really just going to give you the segments that are represented in that particular region of the histogram. Okay, let's talk about mosaics. So I'm going to uncheck my CITO regions, and I'm actually going to unassign it just to get rid of the gray bars for now. And I want to go to my cancer sample that I have up here because I think it's a better example of why we're handling mosaics the way we are. I'm going to turn off a couple tracks to save some real estate. Okay. So we're going to talk about this particular region on chromosome 5. And because we have two separate calling algorithms, one for copy number up here and one for mosaic, you end up with two tracks. So if we were to upload this to the database without doing anything, then this sample would have 7 copy number segments and one mosaic segment representing the same region. And so that's not accurate. It would be redundant. And so we wanted to come up with a way that would be easy in terms of the fewest number of clicks, but as well as best represent the data in the database. So if you go to edit mode, what we're going to do is, in this particular case, for instance, in our last sample, I didn't believe the mosaic. It seemed like a real loss. In this case, the mosaic seems real to me. So this is the segment I want to keep. So in order to do that, I can go back into edit mode, and I'm going to say, promote this segment to the copy number track because this is the one I believe and this is the one I want in my database. So you right click on that segment and say promote to copy number segment. It's going to give me a warning that you're promoting it, and it's going to remove all these other segments up here. Say okay. The copy number state associated with the mosaic will be kept. So in this case, it's saying it's a 2.61 and gain. So okay, great. So now this segment is being promoted above to the copy number. It doesn't retain that light blue. It will go back to a dark blue. But you can tell by mousing over it, we still keep the 2.61 copy number state. So when you're querying in the database, based on the copy number state, you'll still be able to tell that it is a mosaic. So now when I upload this sample into the database, I can see, or I just get one segment representing this particular region, and I was able to choose it to be the mosaic by promoting that particular segment. So again, here on chromosome 8 is another good example where we got lots and lots of the whole integer copy number algorithm is doing its best to make a call on a mosaic. And so as you can see here, you get lots and lots of little segments, but I would be more apt to believe the mosaic segment in this particular case of about 1.46. So again, just to reiterate, we go into edit mode. We would right click and choose promote to copy number segment. It's giving me that warning again that it's going to remove all these overlapping segments, and that's okay. It maintains the mosaic call in terms of copy number state, and it's a loss. So, okay. So, just to reiterate, the copy number segment track, those segments do not get uploaded into the database when you publish. You do have to promote them, those that you want to keep. If you are, for instance, in the last sample, when we deleted the copy number segment that was on the mosaic track, we actually really didn't have to do that in terms of publishing the data to the database because that mosaic track does not get published. So just to keep that in mind, if you have segments on the mosaic track that you just don't want, you want to disregard because you believe the copy number state is more accurate, then you really don't have to do anything with them because they will not be uploaded into the database. It's a good practice to make it as clean as possible, so it's not a bad idea to go in and clean them up, but you won't be putting redundant data in your database if you don't. Right, so then you can publish more than one sample also from the browser if you would like to do that as well. So once I'm finished with this sample, I can take and highlight as many samples as I have loaded into the browser, right click, or just choose publish to database. Files have to be analyzed in HG19 annotation files. So anything that was NA or NetFX 31 and higher, you can publish to the database. Older HG18 files, you would have to reanalyze and in order to upload them into the database. All right, so in this module, we learned how to query the ChAS database and to assign annotations to both the sample and the segments, as well as how to change our database coverage and overlap. Please proceed to the module on reporting in order to see how to export the various graphs and tables from the sample that we just analyzed into the new Word format. Thank you.